Now, some of our rarest and most elusive species are associated with decaying wood and aged bark. But how do we get to grips with understanding their needs, and how do we conserve for them? So to answer some of these questions and to look at this in more detail, first we've got Liam Olds, who's Conservation Officer at Bug Life, and he's going to be talking about, his title is In Pursuit of Rare Saproxidic Invertebrates. Sounds very exciting, and hopefully there's going to be a few beetles in here, Liam. Over to you, please, Liam.
Oh, apologies.
Yeah. So Liam's going to be talking hopefully about some of the things that he's been finding recently with various surveys that Bug Life have been doing at various sites throughout England.
Great, thanks very much, Ross. Just put a timer on my phone so I don't overrun. So my name's Liam Olds, and I'm Conservation Officer at Bug Life, and I'm just going to talk to you about Saproxidic Invertebrates and the survey work we've been doing as part of the Ancients of the Future project for this special group of invertebrates. So as has been mentioned by my colleagues, Saproxidic invertebrates are species that are dependent upon dead or decaying wood at some stage of their life cycle, or they're also dependent upon wood inhabiting fungi or other saproxidic organisms. And they're a diverse group within the UK, so around 2,000 invertebrate species are dependent upon dead or decaying wood in one form or another during their life cycle. And this also includes some of our rarest and most threatened invertebrates in the UK, and some of our most spectacular species as well. And as part of the Ancients of the Future project, we've had 12 primary target species. These are largely beetles, particularly click beetles, and probably the best known perhaps of our target species is the spectacular violet click beetle, which is known from just a few sites in the UK, one of which is Windsor Park, which is a beautiful looking thing. And we also have a few different flies on there as well. So the Royal Splinter crane fly, which has been touched on by Jamie, and also the Western Woodvase hoverfly, which has also been mentioned in previous talks as well. And as well as the sort of primary target species, the rarest of the rare that we really want to survey for and do management work for, there's also a whole sort of suite of secondary species as well. And these sort of saproxidic invertebrates have presented a number of different challenges, and Jamie touched on these earlier, and I'm just going to go into a bit more detail with these. But these saproxidic invertebrates tend to be very elusive, so there tend to be very, very few records in the UK of them. They're difficult to identify, which means you often need experts involved, and it's really difficult to sort of train sort of volunteers to go out and survey for them, because they are that difficult and need a microscope and really sort of specialist knowledge. There tend to be very few known sites for these species. Some of them are restricted to single sites. Some of them, such as the Royal Splinter crane fly, is known from only 2 sites in the world, one of which is Windsor. And they also have very precise ecological requirements as well, very, very niche habitats. And all these sort of factors combine really to sort of result in quite complex survey challenges that sort of require really technical surveys, and they require leading experts in those groups. Another sort of big challenge really with doing a project on saproxidic habitats and invertebrates is the fact that veteran trees don't work to not sort of such short timescales, so they require much longer timescales. So we're not really going to see any sort of measurable change in such a short 4-year project. Regular monitoring is also not really possible, unlike some of the other Back from the Brink projects, because the species are so elusive, because they're so difficult to identify, so rarely seen. And that's where perhaps monitoring sort of the key features, those deadwood features, such as the number of rot holes or the number of sort of veteran trees and that sort of thing, might be more of a better sort of substitute than actually sort of surveying for those actual individual species. And our project, because of these challenges, would really kind of focus more on the sort of assessments of the habitat condition, of the habitat continuity, and also getting that baseline invertebrate data, because invertebrates tend to be very sort of under-recorded. So we know relatively little about them, even on some of these key sites. We've been kind of getting out onto these key sites, and where we think that there's potential for primary species to be, getting out and surveying, not just for them, but also for the wide range of other saproxidic invertebrates as well. One of the sort of target species that we've done surveys for this year has been the Mockus beetle. So this is an endangered Red Data Book 1 species that's legally protected under the Wildlife and Countryside Act. In the UK, it's known only from Mockus Park, which has been touched on earlier, which is an ancient deer park that's home to lots of rarities. And with a lot of these saproxidic invertebrates, we know very little about their larval stages, particularly the larval stages, because they're often hidden away, maybe in underground roots or within cavities in trees and that sort of thing. And we also know relatively little about the adults in many cases as well, and that's the same with the Mockus beetle. But we believe that they inhabit the red rotted interior of ancient oak trees, and they use in burrows and galleries that are made by other saproxidic invertebrates then, and then feeding on other small invertebrates such as mites and colembla, which are the springtails. And so we've been doing targeted surveys for this species, which is one of our primary species as part of the project. And surveys were done between 2018 and 2020, and really with the aim of trying to understand the species distribution at Mockus Park, and to obtain that kind of baseline data really about the trees on which the beetle is found. And the adults are usually found by beating the foliage of thin branches and twigs that are near to rot holes or cavities. And surveys were done in 2018 and 2019 by John Kuta, who's an expert on the Mockus beetle and very familiar with Mockus Park as well. And together with those surveys and all his sort of 40 odd years of survey experience at this site, we managed to confirm the presence of Mockus beetle on 16 veteran oak trees. And there were some surveys done that Alice touched on as well earlier last year, which was partly hampered by COVID, so we could only do 2 days of surveys. But John, who's up on a cherry picker there, and he's sort of up in the tree, and it sort of allowed us really to survey for the species sort of above the normal sort of arm's length that John would normally be restricted to. And that proved quite successful in finding quite a few females. So recorded 26 females, including 19 on a single tree. But unfortunately, the season was really progressed because of the warm weather, and we missed the males, unfortunately. And another species that we've done targeted surveys on is the Western Woodvase hoverfly, which is a critically endangered species that's also found at Mockus Park, and a single site then in the Forest of Dean. And this developed in water-filled rot holes in those ancient beech trees, but also other trees like horse chestnut. And again, this has been touched on earlier, but a survey was done by Andy Godfrey in 2019 that unfortunately only found sort of four females. So we're sort of, well, much lower than previous surveys. So a little bit concerned there, and obviously need a lot more work done on this species to find out how they're really faring at this site. But the survey was successful in finding a lot of other rare and scarce saproxidic invertebrates as well. So that was just a little bit of like a run through about some of the targeted species surveys we've done. But as I mentioned earlier, we've done largely sort of surveys just trying to get that baseline data really to sort of find out what saproxidic invertebrates are found on some of these key sites. And one of the sites that we surveyed is Petworth Deer Farm, which is in West Sussex. A survey was done last year by Mark Telfer, who I'm sure many of you know is basically a legend when it comes to Coleoptera and beetles. And he really focused on the beetles there as a key indicator of saproxidic habitats. And many of these saproxidic invertebrates, as I mentioned previously, are hard to find, and really you do kind of need an experienced entomologist as Mark kind of going out and survey for them and employing a wide variety of different survey techniques to account for all those different species. And Mark has used all of the sort of standard methods that most entomologists would use, so sort of sieving, leaf litter, sweep netting underneath the trees, beating the trees, just directly observing things that might be on the bark and that sort of thing. But then he's also deployed some more specialist sort of techniques that you might use for saproxidic invertebrates. One of those has been the use of vane traps or aerial interception traps. So some of you might have seen these before, but you sort of hang these up within a canopy of trees, or in this case, over a fallen dead oak tree. And what it does is you've got these perspex sheets any sort of saproxylic beetles that are flying through the canopy and flying above the dead wood. They hit the perspex, they drop down through the funnel, and then they collect then in a little sort of jar of ethanol, and they get preserved in there. And these traps are left up for about a week or two, and then the entomologist comes along, collects them, and then can microscope and ID the contents. So as well as this, which is used as a sort of typical standard technique for looking at saproxylic beetles, he also sort of deploys some sort of rather unique sort of methods. And I'm sure many of you are probably getting drawn towards the image on the right, which is an auto catcher, which is something that most entomologists in Britain don't use, a bit more common on the continent. But this is literally just attaching a net to your car. And Mark was driving around Petworth Park, and you're just catching things as you're driving along. And actually, it's supposed to be a good technique for finding a lot of species that you don't normally find when you're out on foot. So that was quite good. And you can just get out the car and attach the net, and then literally just pooter out or collect anything that looks of interest and you want to take back to microscope. On the left there is another technique that Mark used at Petworth Park, which is a subterranean pitfall trap. So you bury these into the ground, anything that's moving through the soil. So if you've got Saproxidic Invertebrates that might be associated with decaying underground roots, they're sort of moving through the soil and all the deadwood material, and then they sort of go through the mesh, and then they fall down into a jar of ethanol again, and then they're preserved, and you can collect them and identify them at a later date. So some really quite interesting survey techniques that was deployed by Mark. And this survey proved really successful. So Mark found 366 species, of which 234 were beetles, and 56 of these species, or around sort of 15% or so, are regarded as key species. Sort of endangered, vulnerable, near threatened, all of those species that are sort of really vulnerable to extinction. And with sort of when you're doing these saproxylic invertebrate surveys, each species has a scoring system. So depending upon how strongly associated they are with particular deadwood habitats, they'll score a value. And the higher the value, the more strongly associated they are with saproxylic habitats. So when you combined all the species from Mark's survey together with all the other species that were already on the site list, effectively Petworth Park comes up with a site quality index value, SQI value of 569.2, which is the 27th highest SQI value in Britain. So that's really good. Obviously shows it's a really quality site for saproxylic invertebrates in Britain. And it's also the third highest in Sussex. And also these species have an IEC value, which is an index of ecological continuity. So this is a value really about how strongly associated they are with sort of unchanged habitats where there's been this continuity of old trees for a long period. So some species will be heavily reliant on really old trees that are perhaps really heavily decayed. So they are really reliant upon old sites that have always had these trees present on site, and others might not be. But when you sort of add up again all of the values from the species, you get one of these IEC values of 87, which is the 17th highest British IEC value. So that's another really excellent outcome, and the highest of any Sussex site. So this really proves then you've got these high quality deadwood habitats that have been present from ancient times to the present. So really suggesting a site of international importance. And also Petworth Deer Park, as a result of all of this, we know that it's unquestionably a site of national importance for invertebrates, and it could well be regarded as a site of international importance as well for its saproxidic invertebrates. So some really good outcomes from this survey. And one of the perhaps more exciting highlights really was finding the false click beetle, which is pictured here, which this was actually only the 5th British individual that's ever been found of this species. So really, really endangered, really rare species. So really exciting to see that. And Mark also had some other quite interesting looking species, and I could be here all day sort of picking out interesting looking beetles. But this particular one is an endangered species. It's a species of rove beetle, a Staphylinid. And it was previously known only from the sort of mid-Thames sort of region, and it lives within the decaying heartwood of veteran oak trees associated with brown tree ant, which is Lasius bruneus. And this was found in one of those subterranean pitfall traps, which I pictured here. So you can see where Mark's have set up is a subterranean trap into the base of this sort of broken oak tree. And this was the first record for Sussex, and also the first record kind of away from that mid-Thames region. So you've had some really, really exciting finds as part of this survey. And just one other one, perhaps less rare than the others, but this is a species of lined flatbark beetle, and this is nationally scarce. It occurs in quite widely across southern England, but it's very rarely encountered. It's flattened because it lives under the bark, and it lives under the bark of beech trees, but also a few other trees as well, such as oaks and hornbeam. And this was found in those aerial interception traps, so those traps that are held up in the canopy and that sort of stuff. But this particular one was in the flight interception trap held over a fallen oak stump. So that's just a bit of a run through some of the species. And just to say, with all of our surveys, then management recommendations would be given to the landowners. So in this case, Mark's recommendations were to keep the existing trees and shrubs, as we kind of want trees to grow naturally and age naturally and die naturally. So minimal intervention to these trees that have these deadwood features is really important. But kind of important things that Mark highlighted was to bring about the end of timber and firewood removal on the site, which is obviously damaging to Saproxidic Invertebrates. To do tree planting to ensure we've got that continuity of habitat going forward into the future. So we ensure we've got our ancients for the future, and also to designate the site as a triple SI as well, because it's not currently, but it's clearly worthy of it because it is a site of national or potentially international importance. So that's me done. So thank you very much for that, bit of a run through, and hopefully that was useful.
The surveys that have been going on lately. Now we're going to move on to a different group of organisms. We're going to be looking at wood rotting fungi. So Chris Knowles is going to be talking about the wood rotting fungi of Great Windsor Park. Chris is a consultant mycologist and ecologist. So we've got 15 minutes now for Chris's talk. We've had a bit of a technical hitch, so Chris is up next, and then hopefully Dave Lammercraft will be after Chris. So Chris, if you'd like to take the stage, please.
Hi, so yeah, I'm glad to get an opportunity to talk about this Back from the Brink project, about the distribution and diversity of wood rotting fungi in Windsor Great Park. I've been listening very keenly to all of the other habitat and species work done over the last couple of days, but fungi haven't been mentioned quite enough, so I'm not going to talk about anything else for the next 15 minutes. So the long-standing royal protections of Great Windsor Park have helped maintain a reasonable continuity of ancient trees in a wood pasture and parkland habitat setting that's quite rare in the UK. This currently means that Windsor Great Park is one of the few UK sites that boasts large numbers of standing dead and fallen veteran trees. The great diversity of wood rotting or saprophytic species supported at Windsor critically relies on the continuity of large volume living and dead wood resources, standing and fallen trees in various states of decay, providing a wide range of niches for different species and the successive stages of their life cycles. This project took the form of a desk study investigating all the known records of non-lichenized fungi found in Windsor Great Park over the last 50 years. Those fungus records were then compared with other habitat data to map the geographical, host and habitat-based hotspots, with all fungus species of conservation importance and those with legal protections. They were all identified, and species whose ecology relied on mature trees and deadwood were all classified separately to be the main focus of the study. Then finally, management and future monitoring recommendations were made in relation to those findings. So how we did it, Windsor Great Park had been historically divided into 21 biological recording compartments, and this project used those compartments for all the mapping and reporting. However, the first step for this desk study was to find the data. Apart from gaining access to the national databases, it was also important to track down all those things in filing cabinets and notebooks by speaking to local recording groups and other individuals who we approached to source any relevant records, all of which were then collated into one single database. In the UK, there are two national databases of fungi, the FRDBI, which is maintained by the British Mycological Society, and Kate2, which is maintained by the Fungus Conservation Trust. Both organisations agreed to share their data with the project. In the end, this resulted in over 33,000 records that were selected as being potentially relevant to the project. However, we couldn't just run a simple filter that would separate all of the ones that we needed out by grid reference or a location name like Windsor, because at least 10,000 of those records had no grid reference whatsoever. And we're using things like localized place names, such as Cranbourne Chase or Cook's Hill and Snow Hill. They were often used, but not always with the same format or spelling. The locations of these records were checked manually by the names of the locations and the features, often having to refer to older maps like this for names that don't appear on current OS maps. A further 7,000 records had a very broad grid needed to be gone through manually by location names for relevance. Once we filtered that through, the list was then checked for valid species names, correcting the many spelling mistakes along the way and cleaning out all duplicate entries. So after coarsely filtering the data in this way, the data set consisted of just over 6,000 records made up of 1,330 species and 582 genera. By comparison, a similar study that was made in 1999 described around 1,200 species having been found at that time. So we're in the same sort of ballpark. This processed data set was then compared against the list of threatened, protected and priority species at national and international levels. Fungal taxonomy, though, has been going through periods of considerable change over the last 50 years. So to ensure that no species records were missed out during the process, we added up to 2 recent synonyms to each species, and then the comparison filter also checked those for inclusion. The taxonomy of each record was also checked at this point, with all taxonomy that already matched the current names given in the Natural History Museum's UK Species Index were left unchanged. However, where records only had older names present, these were updated to current preferred names. And then a secondary data set was created featuring only those species which corresponded with this list of priority species and red lists. After this, at every point another filter was applied or the database was adjusted in any way, care was taken to identify any of these priority species that might be omitted, and notes of those omissions were included in the results. So in the wider data set, 65% of all the records had very poor grid references, making it impossible to successfully map those records correctly. This in turn made it difficult to exactly match individual records to the compartments and habitats on the site. This issue was not as pronounced with the priority fungi records, though, presumably because recorders were more likely to make a special effort to get accurate grid references when they found those. The kingdom of fungi is taxonomically huge. So for the purpose of this study, all the records were divided into 9 loose categories. The categories were divided along a blend of genealogical, ecological and morphological lines, and therefore the lines between them could be quite blurred. So a consistent method of applying categorization to each species was done. And the categories were going from top left, Basidiomycete saprophytes. That's generally mushrooms and bracket fungi, like in that photo, those fungi species on wood. Ascomycete saprophytes tend to be much smaller fungi on wood, as little more than dark spots or dots. And then in the top right, Resupinate saprophytes. It's a category which captures a diverse number of fungi with similar crust-like growth forms on wood. Then on the next row, Ascomycete others is just a huge group of fungi that are generally not typically mushroom-shaped ones. The Basidiomycete other, conversely, is the category that includes pretty much all of your classic mushroom-shaped fungi. Rusts are fungi only associated with the soft parts of the plants. Smuts that utilize the reproductive parts of the plants, particularly grasses. Microfungi is things like molds. And the Myxomycetes, not true fungi themselves, but records of those slime molds are included in the fungus databases. So in actuality, only the first 3 of these categories were relevant to this study. But as none of the, that's because none of the other species or groups have the same reliance on dead wood or mature and ancient trees as a substrate. However, because these databases have now been created, we were asked to map them and assess them as well, which I'll come back to later. So the distribution, as previously mentioned, 46% of the records in the dataset were unmappable. This issue was biased towards only a few genera, and apart from the red listed and priority species, was apparent across the whole set of data, so it was equally balanced throughout. That meant it was still possible to get a reasonable view of fungal hotspots by looking at geographical concentrations of records. Unfortunately, you can see here the map on the left shows the locations of fungus records and how they can be skewed by the use of the lower resolution 4-digit grid references and centralized grid references. So you get a concentration of hotspots on the points where the grid meets and at the center of the grid squares. So on the right, we managed to work with only the higher resolution grid references and the non-centralized grid references us to map distribute to some extent density of the fungal records within each given 1 kilometer grid square, which we could then compare against, to some extent, against the compartments in that area. So using only the records that allowed for the more precise mapping, it was possible to show also host and substrate availabilities and how that was a major factor in either species distribution or the recording bias of people targeting those areas. So using those lists again of the priority species, fungi of Windsor Great Park were checked and filtered to provide the following results. To assess which tree species supported the greatest number of target fungi species, we put together a list of all the substrates and associated organisms that had been recorded across the 6,000 records, which came up with 50 woody plants and trees. However, only 37 of these were host to any of the priority species and red lists. You can see on the wee table to the right there that what it came up was unsurprisingly, Fagus and Quercus stood out as being the most important ancient trees for Windsor Great Park's red listed and priority species of wood rotting fungi. It is also note that the third highest number is on that chart there over at the right, was the number of records that had no detail about associated organisms. So again, it's this disparity in the data that we could use. However, the accuracy of grid references was generally better for these priority species. It was still necessary to only create maps, including records at a 1 kilometer square resolution that could include a good proportion of the priority species. Of the 59 priority fungus species that occur at Windsor Great Park, 10 species either had no records with a grid reference or only 10k square grid references. So putting those limiting factors aside though, there were still clear hotspots in the distribution. You can see on the map here, where that's where the priority fungi, it doesn't match the earlier maps of the fungi data set. So in some areas here, that had very few of the overall records of fungi are quite hotspots for priority ones here, and conversely, some areas that are hotspots had little or no of the other fungi. Other parts of the same studies were compared to the distribution of these against surveys that have been done of veteran trees over 400 years old, and also against percentages of dead wood found in each compartment. However, no clear statistical correlation could be made through that, but the top 10 compartments that had priority fungi in them also were mostly the same top 10 for dead wood availability at the park, and also the same top 10 for trees. So they all overlapped consistently, making up the majority of each combined top 10. So that we did look beyond just the wood rotting fungi. Just briefly cover that we the JNCC SSI selection guidelines and applied them to villages of fungi that we had for tooth fungi associated with the oak, beech and sweet chestnut, and they surpassed the threshold there, suggesting that that assemblage within the park is of SSI quality. Similarly for beech deadwood species and oak deadwood species. More of note perhaps is the grassland fungi. Several of the categories of grassland fungi had high numbers of species diversity, but not all of them, which suggests that further fieldwork and surveys of the grasslands could be recommended. The bottom is not an assessment included in the current guidelines. However, there is a draft available for assessing that assemblage. And Windsor Great Park looked great in that. Species were recorded, so in many ways far surpassing that assessment. However, one of the factors of assessing them is how many of those species were found within the last 10 years. So and the priority species within that assemblage, only 6, so below the threshold, have been found within the last 10 years. Again, suggesting though the ongoing monitoring of the areas where those species are found and of those species in particular would be recommended. So to generally start concluding that, Windsor Great Park is host to a wide diversity of fungi and priority and protected species, some of which were restricted to a very few locations. The large number of records of those species was in part due to the historical continuity of relevant habitats there, but it's also due to concerted efforts made by many mycologists over many, many years. Although the park had a comparably high quantity of fungal records, many of those were actually of poor quality, giving really little of the additional information that would be needed to assess them or to map them in greater detail. In total, this death study found 1,330 species of fungi from 582 genera that were recorded in the park over the last 50 years. Of those, 59 were priority and or red listed species, and 446 species were of the target wood rotting fungi. As you can see in the wee chart here, capturing some of the fungi that appeared on the greatest number of red lists, and 3 of those, the top ones that are pictured. This all reconfirms the importance of Windsor Great Park for fungi. The death study stage of assessments and looking at sort of SSI quality assemblages suggests in it all that it would be advisable to compare current management policies with those that benefit the specific needs and habitat requirements of the 4 assemblages listed here. And I'll go into some of that just now. Further monitoring, for example, as I keep mentioning, the records for the fungi in Great Windsor Park betray a number of biases as to habitats that were examined and surveyed, and a lack of detail common to many fungi records across the whole of the UK. In particular, the lack of quality grid references and notes on associated hosts can be vital for correcting misidentification, resolving taxonomic changes and the like when this sort of survey, this kind of study has been done. So I put forward a suggestion that for all future surveys, an absolute baseline for those surveys and monitoring for professional and just local amateur recorders should be that all their hard-won records should have a minimum of a 6-digit grid reference for all recorded finds, and an 8 or 10-digit grid reference minimum for species that are targeted for monitoring. Also, the associated organism or host should be recorded for all finds. Okay, essentially, all the red listed and priority species should be resurveyed to get up to date and exact grid references, and that will make future monitoring of those species more practical. To round upon Management recommendations, essentially maintaining veteran trees in the woodland parkland would rely on low levels. However, baseline surveys of the quantity and distribution and quality of veteran trees, alive and dead, as well as large pieces of deadwood, should be conducted. Maintaining a supply of deadwood may be vital to the populations. So naturally occurring damage and death will provide enough resources and habitat, so long as the sites themselves aren't allowed to shrink in size, and that the range of trees of different ages are available to become recruited as the new veteran specimens through beneficial management of the surrounding habitats and potential veteranization methods. And where possible, things like paths being rerouted away from those trees. They don't need to be felled for health and safety reasons in the future. Okay, and just to acknowledge all those that helped with the project, are the funders and supporters. That was me, thanks.
Many thanks, Chris. Excellent talk. Fungi, the diversity of fungi is absolutely mind-blowing. I wish I knew more about them, but, you know, there's so many fascinating organisms out there and so little time. So now we're going to hand over to Dave Lammercraft, who's the lower plants champion at Plant Life. Dave's going to be talking to us about can we transplant the rare pox lichen from one tree to another. So Dave, take the stage, please.
Sorry, due to technical issues, you're not getting any slides. So you've just got me talking at you for what might be 10 minutes, I'm afraid. But yeah, so I work for Plant Life as a lichen and bryophyte specialist. I'm going to talk to you about this trial work we've been doing, trying to transplant this pots lichen, Perennial Innitida, at Burnham Beaches. This is work we've done through the H's of the Future project, and I've been working on it with Neil Sanderson, a consultant lichenologist that lots of you will know. So my first slide, if you can imagine, is a beaver and a white-tailed eagle. So lots of, you know, our translocations, we're kind of used to translocations these days as reintroductions. So beaver, white-tailed eagle. I also had a slide of very pretty ladybird spider and lady slipper. So ladybird spider, lady slipper, beaver, white-tailed eagle are all translocations, but they're kind of the reintroduction side of translocation. So that's basically just taking populations back into places where from their former ranges where they've been lost. So with lichens, a lot of what we do tends to be, it doesn't really fit quite as well into those kind of traditional definitions of translocation. It tends to often be sort of rescue attempts. So things like Liberia pulmonaria, the tree lungwort, we've done quite a bit of work over the last few years transplanting that, where, say, a large ash tree on one site, which is very rich, covered in lichens, that came down in a storm. So we took that opportunity to transplant some of the lungwort from that tree to another part of the site, where we lost a lot of the lichens due to dense rhododendron infestation. So that was kind of both a rescue attempt, rescuing the material on the ash tree, but also a reinforcement slash small-scale reintroduction to a part of the site that where it had been lost due to the rhododendron. We've also done some work on a very rare lichen called Bryoria smithii, one of the horsehair lichens. The entire British population of this lichen would probably fit onto the side of an A4 sheet of paper. It's only known on 2 sites in Dartmoor. And I was doing some monitoring work on this a couple of years ago and found a really good clump of it on a dead branch. And it was a good chance to both try the translocation of this thing, which hadn't really been done before, from tree to tree, and also to save that, you know, what would have been an eighth of a side of A4, you know, an eighth of the British population, trying to save that chunk of lichen by transplanting it onto another part of the tree. So they tend to be kind of rescue attempts. With the leafy lichens like the Liberia, they're relatively straightforward to transplant and relatively successful. Slugs are probably the main problem, actually. Okay, so that's a bit about translocations and lichens. So we're talking about Burnham Beaches. Burnham Beaches, I'm sure many of you will know. It sits just to the west of London, between Slough and Beaconsfield. I was whether showing you a lovely old engraving from the 19th century of Burnham Beaches in the Victorian era, which shows back in the mid 1800s this beautiful wood pasture kind of structure with these amazing old beech pollards. And I've also got a photo of Burnham Beaches taken about 2 years ago, and it looks almost identical. There's still lots of beech pollards, ancient beech pollards, and this lovely wood pasture structure as well, which is being restored and managed by the City of London. So the site's an NNR, it's owned and managed by the City of London Corporation, and they're doing a fantastic job of restoring this wood pasture landscape. So Perennial Innitida, it's one of 10 species of Perennial found in Britain. It's a crusto species, so it basically grows immersed in the bark cells. So quite different to things like the Varia pulmonaria, where it's leafy and it sits on the outside of the tree. This grows immersed in amongst the bark cells, so it presents different challenges when it comes to transplanting. It has a very thick, waxy kind of thallus, the body of the lichen, and then I think the name Potts lichen comes from these little black dots that are over the surface of it, which are the fruiting bodies, which are ejecting the spores. It's a southern beech specialist, so it's very much a lichen of southeast England, but it's only known from 2 sites now. Burnham Beaches is one, and New Forest is the other. Relatively widespread, I guess, in the New Forest. But very rare, more of a continental species, I guess, really, but it occupies the kind of native beechwood range in Britain. At Burnham Beaches, was known until we started working there in 2019 from just one tree, a massive old beech pollard, very spectacular tree, but sadly it's dead and being held up by straps, where it's strapped to adjacent trees. And the City of London approached us to ask if we could help with, help sort out this lichen, transplant it somewhere else. It had actually been tried in 2001 by William Purvis, and several fragments of bark were glued onto other trees nearby. And these fragments of bark have actually survived, so they're still present now, 20 years later. But interestingly, they haven't really done anything. So the bark fragments are still there, the lichens are still there on the bark fragments, but the lichens haven't got off of that bark fragment onto the bark of the tree. And there's also no signs of the lichen colonizing elsewhere on the tree. So it's kind of not obviously worked in some sense, but I think the reasons for that being that the bark fragments were glued on top of the bark, so they sit proud of the bark of the receptor tree, and also they weren't really put into the right niche for the lichen. So the idea that we had was basically wanting to try and perform a bark graft in the same way you would do with fruit trees or whatever. So I was trying to think of ways in which we could do this. Someone was working on this with April Windle, who some of you will have come across, just trying out these various techniques. And we were thinking about using grafting wax to kind of seal a fragment in place, but someone pointed out that squirrels would probably love to eat grafting wax, so we ditched that idea. And my favorite idea was to try and take a plug, like a little plug out of the donor tree with the lichen on, and make a similar size plug hole in the receptor tree. And we tried that with an increment borer, which I think actually worked really well for trees that have fairly thin bark, fairly dense wood, and compact kind of plug that you get out of the tree with that. But with old trees, like our big old dead beech pollard at Burnham Beaches, it just doesn't work. It just kind of crumbles into dust, basically. So I resorted to the age-old technique of using araldite to glue stuff in place, and it's a fairly straightforward process, really, of taking a fragment of the lichen from the donor tree, healthy piece of lichen, and we were taking pieces about the size of a 50p piece, And then identifying the niche to where to transplant the lichen into is pretty important, as I said just now. So we were looking for trees with rain tracks, so where rainwater is kind of flushing down the trunk, or wound tracks, so where you get water coming out of wounds in the tree, and where the tree was obviously less acidic, more base rich. So we were using indicator species like orthoticum mosses and the liverwort Metzgeria fleccata, and lichens like Enterographa crassa and Perennial species to kind of give us a clue as to the right kind of niche to put them into. And then it's a case of just digging out a little recess in the receptor tree that's the same size and shape as your fragment you've taken from the donor tree and gluing it in place. So I did have some nice photos to show you how that kind of worked. So we did this in March 2019, and I went back in May of this year to see how they were doing. And some of them had taken really well. So that idea that we were working to, where we're trying to get the fragment to sit into the bark so it's flush with the bark on the trunk of the tree in a kind of natural position, that we do seem to have actually achieved that in a couple of cases anyway. So we also took the opportunity to move some Bellicidia incompta. We found some of that on the ancient pollard. This is another rain track and wound track specialist that likes more base rich bark, a former elm specialist that is quite dependent on ash at the moment. So it seemed quite important to try and transplant this to see if that would work. Has similar habitats to the Perennial, but a slightly different niche in that it likes rain traps and wound traps that haven't yet been colonized by anything. So these little niches get colonized quite quickly by various lichens like the Enterographa and Perennial species and Perennial species. But the Bellicidia incompta needs to get in before those species. It doesn't quite compete. It's got a much thinner sort of body to the lichen. It was quite hard to find the right niche for the Bellicidia because most of these traps were colonized already. But we tried a few of those. So in terms of measuring success, I think one level of success is that you still have your fragments present. So the fact that we've got, I think it's 31 out of the 33 that we did 2 years ago are still there. Quite a few of the ones done by William Purvis in 2001 are still there. So that's a good level of success. The next level up would be, has it spread, has the transplanted lichen spread off of its original bark flake onto somewhere else on the tree? Then the next level up would be, has it done that? And then is it kind of colonizing elsewhere on the tree? And then is it colonizing other trees in the vicinity? And I think there's one example where, I mean, it's early days, it's slightly hard to tell, but I think we might actually be seeing the movement of one of the Bellicidia transplants off of its original flake onto the bark of the receptor tree already. Interestingly, the Bellicidia transplants have decomposed quite quickly, whereas the perennial ones haven't. I think this might be related to the different characteristics of the thallus, the body of the lichen. So the thick waxy thallus of the perennial probably protects the bark to some extent, whereas the Bellicidia has a very thin thallus that probably doesn't. So, I was pretty encouraged by what I saw back in May, both in terms of the survival, the way they were kind of looking nice and flush into the bark of the tree, and with this Bellicidia possibly having spread off as well. That was great. So what have we learned? We've learned that mixing two-part araldite in the field is really messy. It gets everywhere. I got it all over my fingers, all over the trees, all over my notebook, all over my phone, all over the lichens. So it's a bit of a bit messy. So I think using aquarium glue would be a good option in the future. They come in, you can use it with little silicone guns, so a bit more accurate, and you don't have to mix it up and all that kind of stuff. So, and it's quite inert. It's early days, obviously, with these ones that we did 2 years ago, but some of these species of lichens are, they're adapted to colonizing quite quickly. So, I think we might already be seeing the spread of the Bellicidia. The survival of transplanted flakes seems pretty good for the perennial, especially. Identifying the niche is probably the most important thing, and that is quite hard with lichens because there's relatively little research in terms of their ecology. So we're kind of just you kind of have to interpret what you're seeing in the field basically to try and work out where they kind of might want to, when they want to be transplanted to. So and then lastly, I think you can't overlook the fact that the habitat and the habitat management needs to be right, needs to be good for long-term survival of these lichens and their communities. So it's not just a case of we can just pick a lichen from over here and dump it over here and it'll be fine. The management is all really, and that's something that so at Burnham Beaches, actually after we did some of the transplants, the City of London also did some management to open up the light conditions in the wood and make the conditions particularly suitable for the perennial. So can't overlook the importance of habitat management. And that was about me coming to an end. I need to say a big thank you to Neil Sanderson for his help, and to the City of London at Burnham Beaches, who do a fantastic job of managing Burnham Beaches and are very interested in their lichens, which is great. And I was finishing on a picture of a beaver and a picture of Perennial Innitida, just to show you how much more cute the Perennial Innitida is than the beaver, but you'll have to kind of imagine that bit. So there we go, that's me.
Many thanks, Dave. Thanks for that, and thanks for persevering through those technical difficulties. We'll be sharing Dave's presentation, so his slides, after the conference, so keep an eye out for notification of that. Now, if I ask everyone to return onto the stage with me, we'll have a Q&A session. So if you've not already used the live Q&A, a function at the bottom of your screen, use it now if you've got any really pressing questions you want to ask any of the presenters in this last session. I've got a few to begin with, actually for Liam. Now, Liam, I know you mentioned things like the violet click beetle. I remember a long time ago there was a theory that it had an association with birds' nests in cavities in old beech trees. Was that ever found out to be true or not?
I think, yeah, I do think, well, as far as I'm aware anyway, that it does seem to require sort of wood mold. So it tends to be the last thing that colonizes a tree effectively. That's why they're so rare. They need these really old trees that are hollowed out in the inside, then by fungi, and then the contents really basically of all of that broken down sort of decaying material then has got a compost, and then they're sort of within that then feeding on, I believe, other invertebrates and that sort of thing. But it seems to be some sort of requirement that they need sort of bird feces as part of their life cycle as well. And I think that's where the stuff that Steph Skip is doing at the moment with creating those artificial boxes, and then they're using chicken manure as basically a substitute for that. So it's still, I'm not sure if it's fully confirmed, but that's a theory anyway. And it's just, as with most things, you just gotta keep trying different things, and it takes a long time to find these things out.
And is there anything known about, obviously, the trees that they go for, some of these real specialists? What is known in terms of, the tree being in full sun, if it's standing, if it's the humidity levels in the wood? I mean, there's so much to try and figure out there, isn't there?
Yeah, it's really complex. Yeah, and I think as Jamie sort of touched on earlier, there's, you can have, dead wood in so many different conditions, full sun, full shade, high up, low down, wet, dry, it's so varied. And I think working out, you've got to be a bit of a detective really to try to work out the exact requirements of a lot of these species. And yeah, it can take a lifetime of a very dedicated entomologist to try to figure these things out,
Yeah, I think that people don't really realize that, you can devote, decades, even whole careers to try and figure out the lives of even one species. Some of these things are so complicated, and there are so many different things going on. Yeah,
and of course, you've got the different larval stages, and they might have different requirements from adults to larvae, and that's what makes vertebrate conservation so complex. And it's part of the fun of it, I suppose, as well.
Yeah, no, it is. Yeah, because, there's just so much to learn, isn't there? Still, Liam, I'll come back to you in a second. There's a few more questions coming in. Dave, a question for you. Now, do you think lichen translocation should be done more widely, e.g. as a way to restore populations to areas where they've been lost in a way that we're doing with things like red kites and white-tailed eagles and beavers, etc.?
I think it's a tricky one. I think there's a temptation there to think it's an easy way to get these populations back. I think there's lots that we don't know about lichen ecology and how their genetics and their sexual reproduction works and stuff like that. So I think there are questions around just the whole way that lichens kind of work. But having said that, I think there is, we should be looking perhaps at thinking about sort of landscape scale restoration of lichen communities. So it has been discussed with people at Edinburgh, Edinburgh Botanic Gardens, for example. The idea of having a big project to reinstate populations of various species, say, that have been lost. So I think there is an argument for it. I think by and large, though, the key thing really is to get the management right. I don't think the translocation is necessarily a quick win. That makes sense.
Yeah. Okay. And there's also a question for Chris. Chris, with your surveys, you've been looking primarily at fruiting bodies. That's correct, isn't it? Yeah.
Yeah. So it was a dead study. So yes, it's records of fruiting bodies.
And how would you try and sort of extrapolate from that to, because bearing in mind that, often the hyphae under the ground are very cryptic, how can we sort of how can we extrapolate from the distribution of those fruiting bodies to where actually these things might be distributed through the landscape? Because they're going to be much more widespread, aren't they, or not?
Well, no. So for the particular species that we're looking at, or we were focusing on this study, no, you really are limited to sort of real veteran, large pieces of dead wood, oak and fagus. So you can limit that down to only the places where those trees are. For a lot of other wood rotting, I mentioned that we've several hundred wood rotting species that we identified, and a lot of those don't need the tree, the volume of wood or the age of the wood, or even those particular species. And there is there's been some success on grassland fungi using eDNA sampling, which would potentially then allow you to go into dead wood and just take a wee sample and then go and get it sequenced and get a whole list of species that are there, which would answer your query there. But there is issues around loads and loads of the species that you might get there are cryptic species that you don't get fruit bodies from. So for as many as identify a bit of wood that has a fungus on it that would normally fruit, that hasn't been spotted because nobody was there on the right day to see the fruit body before the slugs got it, you're going to find dozens more species that there's potentially no sequence in any gene bank, and a lot of new and tiny microfungi. So yeah, still the best method tends to be field work, going out and looking for those fruit bodies.
And how fuzzy is the taxonomy of the fungi you've been looking at? I mean, because reading some things, I mean, obviously with insects that I'm aware of, taxonomy is often quite fuzzy, but then fungi seem to be a whole another level of fuzziness in terms of what we deem to be one particular species is actually much more diffuse and complicated than that. Is that?
Yes, a lot of species complexes. Yeah, so that's coming up. The whole of fungal taxonomy is in a state of constant flux, I think. Has been for some decades now, but with the advance in sequencing and stuff, it's really changing the game. And it's somebody can discover that what we believed was a single species might be half a dozen species, but it doesn't really mean anything unless you can match that to morphological or micro characters that you can actually say, well, this is where the line is between this species and that. So, but yes.
And will you be able to use things like the handheld sequencers like the mini iron and things? Do you reckon you'll be able to use things like that in the field in the future or not?
It seems to be the way that everything is going. We're just at the moment I just don't think there is enough eDNA sequences in the banks to cover all of the. Either there isn't enough, or they're not all reliable. The ones that are out there at the moment. So there's just too many unknowns for it to be efficient. But I'm sure that we'll get them all eventually, and it'll become more and more relevant.
Okay. Well, thanks very much to all three of you for some fascinating talks.